All right, I'd like to welcome everyone. I'm Sandy Williams, Gladstone president, and as this overflowing auditorium attests, we've gathered here today for what really is a singular highlight of every year at Gladstone. Once a year, we invite the most pioneering and accomplished scientists of the planet to come and join us for a day and to share with us their experiences and help us learn some of the ingredients of what goes into a career of great success in science. I'd ask all of you to look at your program and look at those that have preceded Jennifer Doudna in this series, but Jennifer certainly adds luster and distinction to what has been a great a great tradition. I'd also like to point out that the event today honors Lloyd Holly Smith. Many of you know this, but some of you may not. Holly Smith is one of the great figures of American academic medicine. He is, without question, one of the handful of people that did the most to drive the great ascent of UCSF into the stratosphere as one of the leading academic medical centers in the world. But his influence on American academic medicine was actually far greater than that. It reached out all over the country where at least one generation and likely two generations of scientific and medical leaders who followed learned from Holly and had him as a personal friend and mentor. I count myself in that group, and I know many of you do as well. So we're here to honor Holly. Holly and his wife Margaret would be here with us, except for the fact that a few days ago, Holly underwent bilateral knee replacement surgery, which at the age of 93, I think, was designed to get him back on the hiking trails in Marin. So we're counting on Holly to be here in person next year. Also, I need to honor Patty Fitzpatrick, who's here in the front row. Patty has graciously given us the gift that's endowed this lectureship and allowed it to go on. But she, too, connects personally to many of us. Her father, Jim Weingarten, was my first department chair, great friend and mentor to several others of you, and later became the head of the National Institutes of Health. So, and Holly and Jim go back all the way to the days of their internship as friends. So there's a lot of family that's represented here today, and I wanted everyone to know that. But now on to the main event, and I'll ask Warner Greene to introduce our most distinguished guest.
Thank you, Sandy, and welcome. Let me add my welcome to all of you. It's quite a crowded house today. Today is, in fact, a signature event for Gladstone. Our annual distinguished lecture named after Holly Smith is one that we look forward to with great anticipation. And I must say, though, that this year's honoree, Professor Jennifer Doudna, has especially excited us with, as she will be talking about a technology that has revolutionized modern biology. I don't think it's an overstatement to say that she is one of the most sought-after speakers and important scientists in the world today. Now, I would point out that it was in January, I have the flyer to prove this, January 26, 2012, that we invited Jennifer to give a talk here in the Gladstone Institute of Virology and Immunology lecture series. She just said, I think I want to talk about something new. And the title of her talk was Dicing and Beyond, Regulatory RNA in Humans and Bacteria. And we think, we're not quite sure, we think this was probably her first public presentation of the CRISPR-Cas9 studies. And I remember her seminar. I sat in the kind of in the right front, listening and thinking, wow, this is pretty cool. I wonder where this will go. So, you know, now that was, remember, President Obama had just been appointed to his second term as president when this whole thing took off, and it's been, I would guess, a rocket ship since then. Let me tell you a little bit more about Jennifer, and then we'll let her, why you came. Jennifer took her undergraduate degree from Pomona, her PhD from Harvard, working with Jack Szostak. She went on to do a postdoctoral fellowship with Tom Check at the University of Colorado, and then became an assistant professor at Yale University and joined the Howard Hughes Medical Institute. In 2002, she moved to Berkeley. She continued there as an HHMI investigator, and she holds the La Ka-shing Chancellor's Chair in Biomedical and Health Sciences, and she's also professor of molecular biology, cell biology, and chemistry at Berkeley. She's won numerous prizes. Let me just mention a few: the Waterman Award, the Lurie Award, the Janssen Award, the Breakthrough Prize in Life Sciences, the Gruber Award, the Mastery Prize, the Gardner Award. There are many, many more. I could continue. She was elected a member of the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Inventors, and a foreign member of the Royal Society. She was also named as one of Time Magazine's 100 most influential people in the world. Today, we're pleased to welcome her to give her lecture entitled CRISPR Biology, Nature's Toolbox for Genome Editing. Please welcome Jennifer Doudna.
Thank you very much. Well, good afternoon, everyone. That really an incredibly wonderful day I've had here. Thanks to all of my colleagues and the students and postdocs that met with me today. Really, it's been very stimulating. And thank you, Warner and Sandy, for that generous introduction. It's really an honor to be here back at the Gladstone, right across the bay from where I spend much of my time. And the story I wanted to tell you today, I wanted to do a couple of things. I wanted to start off by talking a bit about how I got into the area of biology that led us to gene editing technology. And you'll see that this is largely a story of serendipity in science. So certainly for all the students in the audience, this is, I think something that for me at least has been one of the most fun things about my career in science. Is just sort of the unexpected nature of the things that we do. And then I wanted to tell you about some of the work that we're involved in, but obviously many others, in thinking about how we're going to apply gene editing, especially as a therapy for people eventually, and then also what we're thinking about in terms of where this technology and all of the things that it's spawning are going to go in the future. And then I want to end by saying a little bit about the ethics of gene editing, something that I've gotten really engaged in over the last couple of years. So I wanted to start off by just pointing out that I think ever since the discovery of the structure of DNA, scientists have really been curious about sort of this question right here. What if it were possible to make precise changes to the letters in the DNA code? And for a long time, this seemed like a sort of a fantastical proposition. But then over the last 10 years or so, technologies have come along that have enabled scientists to do exactly that. The challenge was those technologies were not very easy to use. And so I think the field, in a way, was kind of ripe for an opportunity to do what we now call gene editing in a way that would be simple, fast, and accessible to a wide number of people without people that didn't have to have particular expertise to do it. And the really interesting thing about CRISPR technology is that this is a technology that came about not through a focused effort to discover it, but rather through a project that was very much small science, and science that was pointed in what was seemingly a very different direction initially, which was namely trying to figure out how it is that bacteria are able to fight viral infection and do that using small RNA molecules, something that I had sort of gotten interested in due to my long-standing interest in the function of RNA in the control of genes in cells. And so for me, my first awareness of the fact that bacteria might have an adaptive immune system actually came about from a conversation I had with Jill Banfield, a colleague of mine at UC Berkeley. And, in one of the first sort of serendipitous twists to the story, Jill works in a different college than I do. She's not an experimental scientist. She does computational biology, and her lab studies bacteria by investigating the nature of their genomes and doing metagenomic sequencing, and then asking what we can learn about populations of bacteria that have never been cultured in the lab by simply looking at the sequence of DNA that makes up their genome. And through doing that kind of work, her lab and just a few others about 10 years ago had realized that a lot of bacteria have in their genomes, this is a cartoon of the circular DNA genome of a bacterial cell, many bacteria have a repetitive DNA sequence that occurs in the genome and consists of a series of direct repeats. These are 40 base pair, roughly 40 base pair DNA sequences that occur over and over. And in between those sequences are unique sequence elements that in 3 papers published in 2005 were shown to derive from viruses, the viruses that infect these cells. And this was actually the first hint that what these arrays of sequences might represent was some sort of adaptive immune system in bacteria, a way that bacteria could acquire DNA from the viruses that infect them and then perhaps somehow use that information to protect the cell from future infection. And what made it seem like sort of a system that bacteria had evolved to fight viruses was the fact that in addition to these arrays were sequences known as so these had come to be called CRISPRs, and close by in the genome were typically CRISPR-associated or Cas genes that were maintained together with the arrays. So it really had the look of some sort of a system in bacteria. And so Jill told me about these and thought that it might be very interesting to investigate whether these sequences were being employed at the level of RNA. In other words, the cell might make a little temporary copy of these loci in the genome and then use that genetic information in the form of RNA to find and destroy viruses. And this was a hypothesis, no evidence for this at the time. And what emerged over the next several years from work that was done in part by scientists that were at the time working at a yogurt company, Rodolphe Berengo and Philippe Horvath, as well as some biochemical scientists in the Netherlands, and Blake Wiedenheft, a postdoc who came to my lab around this time to start working on these systems, was that, in fact, bacteria that have a CRISPR array are able to protect themselves from viruses using a pathway that involves detecting viral DNA that gets into the cell, such as from a viral infection, and then integrating little bits of that DNA into the CRISPR array such that the cell can make a copy of it in the form of RNA. And then these RNA molecules are chopped up into units that each include one sequence that comes from a virus, and then those RNAs assemble with proteins encoded by the Cas genes to form RNA protein complexes. These are essentially surveillance complexes in the cell. They search through the cell looking for DNA sequences that match the sequence in the RNA that comes from a virus. And then when that match is found, these complexes are able to latch on to the DNA and cut it up. So let me show you in a little bit more detail how these bacterial immune systems operate. So this is a cartoon of a bacterial cell with a genome containing a CRISPR array. And as I mentioned, these are copied into RNA. And initially, these molecules of RNA are made as one long stretch of sequence that includes multiple virally derived segments. And then the CRISPR system typically has an enzyme, a protein that will chop these apart into the functional RNA units. These each include one virally derived sequence that's flanked by a copy of the RNA of these DNA repeats on either side. So each RNA molecule has a unique sequence in the middle and identical sequences on the end, so that these function really as handles that allow them to assemble with the Cas proteins to form surveillance complexes that can recognize foreign DNA and lead to its degradation. And so in my lab over the last, ever since that first conversation with Jill Banfield, I thought this sounded really crazy and really kind of cool and interesting. And so I started working on it just with really just one or two people that came to the lab, Blake Wiedenheft and then Rachel Horowitz, who came to the lab to study this process. And we've worked on essentially all the aspects of this, including how new sequences are integrated into the CRISPR array, how these RNA molecules are produced and then assemble with proteins, and then how DNA targeting works. And today what I want to focus on is really this question here that we set out to investigate, which is how RNA-guided DNA cleavage works. Because in the end, this is really the heart of how these CRISPR systems operate, is they function as RNA-guided DNA targeting systems. And one of the things that's really cool about CRISPR biology is that it emerged early on that there's not one flavor of these in bacteria. There's actually many different versions of CRISPR systems. This is actually a figure taken from a recent paper published by a review article published by Eugene Koonin and colleagues. And what's currently understood is that we can sort of roughly classify CRISPR systems into these two classes. Class 1 up here that each consist of CRISPR systems that include multiple proteins encoded by these genes that are required to form the targeting complexes with the RNA. And then down here, the class 2 systems that each include a single protein that is an RNA-guided enzyme that is responsible for protecting the cell. Over the years in the lab, we've worked on all of sort of different members of these different classes. Started off initially studying what are known as the type 1 systems up here, and then it was really through a great collaboration with Emmanuel Charpentier's lab that we started working on what are known as the type 2 systems down here, in which a single enzyme called Cas9 had been genetically implicated to be responsible for viral protection in cells that had this type of CRISPR system. And so when Emmanuel and I got together in 2011, we decided to put together the expertise in our two laboratories to figure out the answer to the question of what the function of this Cas9 protein might be. And so this led to a really fun collaboration that occurred across the ocean and involved two terrific people in our labs, Martin Jinek, a postdoc in my lab, and Krzysztof Chylinski, a student in Emmanuel's lab. And what these guys figured out was that Cas9 is a really amazing enzyme that's shown in this cartoon as the blue blob. It's an enzyme that whose job is to find sequences in DNA that match a 20 nucleotide sequence that's present in a CRISPR RNA. So remember the picture I showed you of the CRISPR array, and a little RNA copy is made, and then that RNA binds to the Cas9 protein and provides the targeting information that tells this protein which DNA molecule to latch onto. So when a sequence of 20 nucleotides in the DNA matches this 20 nucleotide sequence here in the RNA, which would originally be coming from a virus, then this protein is able to recognize that place in the DNA, unwind the DNA, and two enzyme active sites cut each strand of the DNA double helix. So it makes a clean, double-stranded break in the DNA. And in bacteria, this then leads to destruction of that DNA molecule. So for finding and destroying viral DNAs, this was a very effective system. Now, we were doing biochemistry on this. So that means we were working with purified proteins and RNAs and trying to really figure out the molecular basis for this DNA targeting activity. And what Martin and Krzysztof figured out was two things that turned out to be really fundamental to how this works. One is that they found out that this system requires a second RNA molecule known as tracer, this molecule right here, that Emmanuel's lab had previously shown to be important for production of these mature functional CRISPR RNAs. And Martin and Krzysztof found out that this RNA actually sticks around and forms a binding site by interactions on the end of the CRISPR RNA that is essential for Cas9 protein binding. So this is a handle here that allows assembly with the protein. So you have to have both of these RNA molecules together with the protein for this DNA targeting to work. And the other thing they found out was that targeting required not only this match to the 20 nucleotides in the CRISPR RNA, but also that this DNA, this sequence in the DNA be adjacent to a little motif, a little sequence motif in the DNA called the PAM, which for this Cas9 enzyme is a GG dinucleotide, 2 Gs in a row. And by following those rules, we found out that we could actually program this protein to recognize and cut DNA sequences by simply changing the sequence of this short stretch of RNA in the CRISPR RNA right here. And it was really from doing those experiments, there was a sort of a fateful day when Martin Jinek was in my lab, in my office at Berkeley, and he was showing me all of his biochemical data. And we were saying, wow, it's really crazy. This is like a programmable protein. You can program this thing, and it'll go find a DNA and cut it. And we realized that we could actually simplify it compared to what nature had done by linking together the two RNA molecules into a single RNA. We called this a single guide RNA that would have the Cas9 binding structure over here and the targeting sequence on the other end. And then by simply changing this short end sequence on the end of this RNA, we could program this enzyme to find and cut DNA sequences that matched this sequence element right here. And so we really turned this into a two-component system, a single RNA, and of course the single Cas9 protein that could be programmed to generate double-stranded breaks in DNA. And this was the idea. And so Martin ran to the lab to test this, and he basically made a series of 5 different single guide RNAs that would recognize sequences found in a circular plasmid DNA molecule. And we're doing these experiments in the test tube, so we have everything is sort of a purified system. We could figure out what minimal components were necessary for this programmed DNA cleavage. And these sites were chosen simply to be adjacent to a GG dinucleotide in each case in the DNA. And then by incubating the Cas9 protein with one of these guide RNAs and a restriction enzyme that cuts the DNA a short distance away in the plasmid, we could analyze the products on an agarose gel system. And you might be able to see here maybe these little fragments of DNA that are released after digestion that each corresponded in size to the size expected based on the cleavage site directed by these single guide RNAs. And this was really for us kind of the moment when we realized, wow, this is not only a cool protein, but it could be a really exciting tool, a technology. And just to show you in a little more detail how we understand that this protein works. So this is a 3D printed model of Cas9, sits in my office at Berkeley. It's about this big. And it's based on a crystallographic structure of the protein that was solved by Martin Jinek and his lab members at the University of Zurich, where he's now on the faculty, and a model that was put together by Jacob Korn at the Innovative Genomics Institute at Berkeley and UCSF partnership. And what you're seeing here is the protein, which is in white, holding on to its guide RNA in orange and interacting with a double-stranded DNA substrate, the blue molecule. And you can see that inside the protein, when targeting occurs, the protein unwinds the DNA strands so that an RNA-DNA interaction can occur, the orange and blue helix that forms inside the protein. That's really the targeting mechanism. And then 2 active sites are activated to cleave the 2 strands of the DNA. And in my model, it's actually, these are held together by magnets, so we can actually pull this apart and look at what happens after DNA cutting and this DNA molecule is released from the protein. So it's a great programmable protein. It makes double-stranded DNA breaks, but why is this interesting as a technology? And this is where it was great that, every week or so, at that time, journals were landing on my desk or showing up in my inbox that had articles about a seemingly unrelated field, which was the field of genome editing, and scientists that were making programmable proteins that could do this, that could cut double-stranded DNA at particular sites. And these were proteins like zinc finger nucleases and talons, very interesting as tools, and we were sort of evaluating them in our own lab thinking, gee, do we really have the expertise to use them, and had decided that we didn't. But we realized when we saw the activity of this bacterial enzyme that it actually has a capability analogous to these types of programmable proteins, zinc finger nucleases and talons and homing endonucleases, in which double-stranded DNA breaks can be introduced into DNA. And if this occurs in the context of a cell, like an animal or a plant cell, those types of cells have very sophisticated machinery for repairing double-stranded breaks. So unlike in bacteria, where double-stranded breaks are typically lead to rapid destruction of the DNA, so useful for targeting viruses, in plants and animals, these breaks are found and repaired, and they're repaired in pathways that include something called non-homologous end joining, where the DNA ends are pasted back together chemically, sometimes with a little disruption in the DNA sequence at the site of repair, or they can be repaired by a process of homologous recombination, in which a DNA molecule is actually integrated into the broken DNA in the process of repair. And so many people had appreciated that you could trigger targeted site-specific DNA editing if you could introduce a double-stranded break into the genome at a particular site. And the question was how to do that. And so these earlier technologies were effective, but they were difficult enough to deploy because they involved protein engineering for every experiment, such that most labs had not been able to really access them. And with Cas9, the exciting thing here was that a single protein could be trivially reprogrammed for different DNA recognition and cutting activity by simply introducing a desired sequence into this single guide RNA. And so that was the idea. And so let me show you a video that just was made for us by a wonderful artist at University of Utah, Janet Iwasa, and illustrates the way that we imagine that these bacterial enzymes operate when they get inside of an animal or a plant cell. And so this shows, we're zooming into a cell, and of course in an animal or plant cell, the DNA is inside the nucleus. And not only that, the DNA is packaged into chromatin, and what you see here is the blue DNA wrapped around green histone proteins. And somehow this bacterial enzyme Cas9 with its guide RNA has to search through all of the DNA in the genome, packaged in this way, to find a single site that matches the sequence found in the guide RNA. When that interaction occurs, the protein holds onto the DNA and unwinds it to form this RNA-DNA hybrid, cuts the DNA, and then hands off these broken ends to the repair machinery in the cell that, in this example, shows integration of a DNA sequence in the process of repair. And so when you look at something like this, it looks really fanciful, and yet we understand that somehow this bacterial system is very effective at doing this kind of DNA cleavage, leading to site-specific repair in virtually every type of cell and organism where it's been tested. And so in my own lab, over the last 4 years, we've been really interested in figuring out how that works and why it works, and then how we can address some of the challenges to using this technology for applications in human medicine, but also in other systems like in agriculture. And so let me just tell you a few things that we've learned about how this system actually operates inside of a cell. And so one of the questions that we wanted to address was what drives DNA target recognition. And this is a cartoon that again shows what happens when Cas9 with its guide RNA binds to a matching sequence in a DNA molecule. And we know that this triggers DNA unwinding kind of locally, sort of melts out these base pairs in the DNA so that one strand of the DNA can make base pairing interactions with the guide RNA. So this is the RNA-DNA helix that I showed you before, right here. And then that triggers activation of the catalytic activities of the enzyme, leading to DNA cutting at these sites. And this is all occurring right next to a little motif called the PAM. And amazingly, this is an enzyme, this is a protein, that is able to unwind DNA without any external energy source. So it doesn't hydrolyze ATP or GTP. Somehow it gets the energy for melting out and opening up this DNA duplex from somewhere else. And so we've been sort of really intrigued by this and interested in understanding how that works. And I just want to share with you a few things that we've learned about this process. The first of which really came from experiments we did with Eric Greene's lab at Columbia using DNA curtains. And so these are experiments that involve taking molecules of DNA, and in this case these are 48 kb phage lambda DNA molecules. And for those of you that don't know what that is, it basically just means these are bacterial viruses. They're the viral DNAs. And so what you're seeing here are each strand of this DNA curtain is a virus genome that's been appended to the slide on one end and then visualized using a green dye. And we can extend the DNA by flowing buffer across the slide from top to bottom. And when the buffer flow is turned off, you see these DNA molecules just coiling up to the origin. And then we can take the Cas9 protein and label it with something called a quantum dot that allows visualization of Cas9 as it interacts with the DNA using a technique called turf microscopy. And you can see in this example that a lot of these Cas9 proteins align on the DNA strands at a position that they've been programmed to recognize right here by the use of a guide RNA that recognizes that sequence in the DNA molecule. And so this was in a series of experiments done by two students, Sam Sternberg in my lab and Cy Redding, who was with Eric and is actually now a fellow here at UCSF. These two guys teamed up to do experiments that revealed, I think, three sort of fundamental conclusions about the way Cas9 operates. We found out that DNA binding begins at the PAM motifs. So rather than starting with interactions between the guide RNA and the DNA, it actually starts with interactions at these little PAM motifs. And then somehow that contact between the protein and the PAM triggers DNA unwinding, allowing hybridization with the RNA and activation of the enzyme so that it can actually cut the DNA. And then finally, we found out that the protein, it has a very high affinity for the DNA product, and that means that after it cuts the DNA, it stays very tightly bound to the broken ends of the DNA, such that we saw no substrate turnover. In other words, for every molecule of Cas9 in these experiments, we got one DNA molecule cut. And so, but again, thinking about how this actually operates, how it works, we were interested to understand from a molecular structure perspective how this protein is able to interact with DNA and unwind it. And one of the clues that we got to how that might work came from work that we did with Eva Nogales and her lab members at UC Berkeley using initially just very low resolution electron microscopy, not cryo. This was just negative stain electron microscopy, looking at Cas9 protein alone and then the protein bound to a guide RNA and then the guide RNA and the DNA being present. And what you can see in this cartoon is that we could see even at very modest resolution that the protein structure changed dramatically upon RNA binding, such that a channel opened up in the center of the protein where the RNA-DNA hybrid winds up once this complex binds to a DNA molecule. And now that we have, we collectively, the community, has access to a whole collection of crystallographic structures of Cas9, it's been possible to see what this conformational change in the protein is all about at much higher resolution. And this is a little video that was put together by an undergraduate student at Berkeley, Aubrey Thompson, that shows morphing between the different crystallographic states of Cas9. And as you watch this, you'll see the movie starts off with Cas9 protein alone, and as it morphs to the structure bound to the guide RNA in orange, you saw a big rearrangement of RNA and DNA molecules look like inside the protein. The RNA is orange and the DNA is the magenta. And in these recent structures that we solved using both crystallography and electron microscopy, we can see the open DNA duplex and these two sites in the DNA that are actually cut by the enzyme once this complex forms. So it looks like a protein that has really been evolved over, probably eons in bacteria to be highly regulated, so that it really doesn't assume its active structure for DNA cleavage until and unless it's engaged with both a guide RNA and a matching DNA molecule. And just to show you in a little bit more detail, this is actually a recent crystallographic structure that we solved of the Cas9 protein bound to the green guide RNA, in this picture green, and the magenta DNA. And what you can see here, different from the model I showed you before, the 3D printed model, is that now with this double-stranded DNA substrate present in the protein, we actually see that this active site has swung into position to cut the DNA. So it's like a little arm that swings down and then actually cuts. So then thinking about the behavior of this protein, so I want to just just loop back to what I said about the energy of DNA duplex unwinding. We suspect, although we don't really have conclusive evidence for this, that it's really coupled to these conformational states of the protein, and that the structural changes in the protein are what help trigger DNA unwinding and provide the energy to begin melting the DNA duplex and allow interactions with the guide RNA. And then you could ask, well, how does this, that's fine in a sort of in a biochemical experiment, but how's it working when it's really inside of a living cell? And this is a model that we had put together based on data from the work with Greene's lab, so in vitro single molecule experiments with these individual DNA strands of lambda DNA, in which our data were consistent with the idea that Cas9 and its guide RNA are able to bind and release DNA very quickly. So our data suggested that rather than grabbing onto DNA at one end of the genome, let's say, and trying to slide along to find a target site, this protein instead operates by binding and releasing DNA very quickly and kind of randomly, but slowing down when it encounters one of these PAM motifs to allow interrogation of the adjacent DNA sequence. And if there was a base pairing that could occur between this DNA strand here and the guide RNA, then that triggered initial DNA unwinding, leading eventually, as long as this strand sequence still matched the sequence of the DNA, to opening up of the DNA and eventually activation of these catalytic sites to generate the DNA break. And we've actually started to do experiments that would ask how this operates inside of a living cell. And I'll just show you one slide about this, but this has been a really great collaboration with the lab of Robert Tijan at Berkeley and his colleagues at Janelia Research Campus, and a wonderful chemistry student, Spencer Knight, who came to the lab to do this. And what Spencer did was to generate a cell line, mouse cell line, that had a fluorescent label on Cas9. And what you see over here on the right are individual particles of Cas9 as they move around the live cell nucleus. So you get a sense of just how rapidly these particles are interacting with the DNA in the nucleus, even though it's a very compacted environment. And then what we can do to probe this system experimentally is to program Cas9 with different guide RNA sequences and ask how those different guides affect the kinetic behavior of the protein. And over here, you can see on the top that if we use either a nonsense guide that doesn't recognize any sequence in the genome, or a guide RNA that has mismatches adjacent to the PAM sequence of a target site in the DNA, we see very rapid kinetic behavior of the particles. But if we program Cas9 with a guide RNA that has mismatches only further away from the PAM or has a perfect match to a sequence in the genome, and by the way, this is a very abundant sequence that occurs about 200,000 times or more, you can see that a lot of the particles now have much slower kinetics, and we attributed this to binding and sort of long-term binding and residence time on these DNA target sites. And again, from doing these kinds of experiments, our conclusion was that the search mechanism of Cas9 in these live cells really involves three-dimensional diffusion rather than processive interaction along the length of the DNA. So we think that one of the ways, so a lot of people have asked, well, how does this bacterial enzyme deal with the compacted DNA in the context of chromatin inside of animal and plant cells, something that it would not have presumably had to deal with over evolutionary time? We think that it's actually a serendipitous property of the enzyme that it has this ability to bind and release DNA very quickly, and thereby maybe take advantage of transient dynamics of chromatin in these cells. So how do we think about using this kind of technology therapeutically? And I really think that, beyond sort of the immediate questions of targeting accuracy, something that I think has largely been addressed in different ways by the community, and I'm happy to answer questions about that later. I think the current bottlenecks are really these three right here. Delivery, how do we get these molecules into tissues in a selective way? How do we control the way DNA is repaired after it's cut? And then how do we address the ethical challenges that come up with certain kinds of applications of gene editing? And in humans, that would probably largely be editing that would occur in the human germline. And so today I want to just tell you a little bit about work that we're doing in the lab across the bay to address this issue right here. And one of the things that we've been thinking about, and systems we've been thinking about, is how you might introduce these editing molecules into particular tissues or organs. And this really started for me because I had a wonderful postdoc come to the lab from Stanford, Brett Stahl, who started working in the lab right after we had done this initial work on Cas9. And he was really particularly interested in addressing Huntington's disease, which is a disease in which there is an expansion of a particular short triplet sequence in DNA in a particular gene encoding the Huntington protein, and leads to a neurodegenerative disease that often strikes people in their middle ages and is really a devastating thing. And so we wondered whether we could develop ways of using the CRISPR-Cas9 technology to correct or at least mitigate this genetic disease. And so this started as the work of Brett Stahl, and we had a great collaboration that's ongoing with Roche Biopharma to address this. And then we have a new postdoc, Alex Amen, that's coming to the lab to work on this, and it's been an expanding project that now includes Mitch Berger and his team of neurosurgeons at UCSF. So we're really excited about some of the data that we've been getting and opportunities to expand this to address other challenges, disease targets in the brain. And so just briefly to tell you about some of the work that Brett and his colleagues have been doing in the lab. So we started thinking about ways that we might be able to deliver this protein and its guide RNA into cells. And of course, many labs have been thinking about this, and so there are various ways to do this that involve genetically encoding the protein and its guide RNA. So you can use DNA vectors, plasmids to do this. You can encode them in viruses. You can use messenger RNA that encodes the protein, and you can encapsulate that in lipid particles if you want to. And so these are some of the strategies that people have used. But we're biochemists, and so we actually started thinking about using materials that we were actually making every day in our lab for experiments that we were doing. And we started asking, gee, I wonder if we could just deliver the purified protein and a guide RNA directly into cells. And so the strategy would be to take what we call RNPs, RNA protein complexes that are pre-assembled, and introduce them into cells. And the idea was that maybe we would observe fewer off-target effects because of the shorter lifetime of these pre-assembled protein particles and no foreign DNA that's going into the cell. So no chance of integrating the gene into the cell's DNA. We wondered if we might see a lower immune response to these particles, and also whether we could think about ways of engineering cell type specificity to the delivery strategy. And so I just want to tell you a little bit about work that Brett has been doing along these lines, and initially was helped a lot by another postdoc, Steve Lin, who's now gone back to Taiwan and is running a lab at Academia Sinica. Brett and Steve purified Cas9 and started adding guide RNAs to form these assembled complexes, and then using them to edit cells, different types of cells in the laboratory. And one type was these neural progenitor cells. And we knew we wanted to eventually be able to actually test this in animal models. And so Brett's idea was to actually take advantage of a reporter mouse that had been created called the TD tomato reporter. So it's a mouse that has a gene encoding a red protein, red dye, that's integrated in the genome. And it's integrated, here's the TD tomato gene, it's integrated in such a way that there are sequences upstream of the gene that prevent its expression unless they are removed. And there are different ways to remove them, but Brett's idea was to actually do this using Cas9, where you could direct Cas9 with guide RNAs that would bind to these parts of the DNA, and through genome editing, remove the sequences that block expression of the tomato gene. And the great thing about doing this is that if it works, you actually get expression of a red dye in cells so that it's very easy to screen visually and observe cells that have been genome edited. And you know they've been edited in the right place because only changes at this site will trigger expression of this gene. And so Brett found that he could actually do this with these neuroprogenitor cells in the laboratory by simply treating the cells in such a way that they would take up these particles. But then the question was, how are you going to actually do that if you wanted to do it in an animal brain? And so the idea was to actually do this. Let's see, I think I have one picture here that just shows how this works in these neuroprogenitor cells. And so this is showing an experiment where we use different amounts of the Cas9 RNA protein complex, ranging from none, so this is a control over here, up to 100 picomolar and some intermediate amounts. And you can see that you start to see red cells showing up, and you can actually sort these in a cell sorter, and you can quantify how many cells remain unedited versus those that receive an edit. And then, Brett was able to also look at this by actually sequencing the DNA in this part of the genome and show that these edits had been introduced as expected. But then how are we going to get these into animals and into animal brains? And so we were sort of inspired by work that Carlos Barbas's lab had done some years ago with zinc finger nucleases, where they had actually introduced cell-penetrating peptides onto zinc finger nucleases, which are programmable proteins that can also trigger genome editing. And so we thought we would try this with Cas9. And so Brett's strategy here was to introduce different numbers of these peptides that are abbreviated NLS for nuclear localization signal. These are just basically peptides that are charged and can be placed onto the end of the protein sequence, along with a green fluorescent protein that gives the protein, the Cas9 protein, a green color, so we can observe its ability to get into cells visually. And this is just showing an experiment that we did, that Brett did in the lab, where he is able to introduce these modified versions of Cas9 with the cell-penetrating peptides onto cells. And the experiment was really to ask, if we don't do anything special to the cells to allow them to take up Cas9, is there a benefit to having these cell-penetrating peptide sequences appended to the protein? And what you can see over here, and I think there's one more, yeah, there's a slide right here. But basically what we found, if you want to look down here, these are assays that we use to test for gene editing. And this is a control experiment where we take cells and we treat them using something called nucleofection that allows them to take up even unmodified Cas9 proteins. And you can see that in this experiment, we have a lot of gene editing that's going on, signified by the presence of these DNA products right here. But if we do experiments where we use basically direct delivery, which means we just add this RNA protein complex to cells, no special treatment, no nucleofection, if we do this experiment with a Cas9 protein that doesn't have any cell-penetrating peptides on it, we see really no evidence of these DNA-edited DNA products over here. But if we do this with Cas9 protein that's been modified with cell-penetrating peptides, we actually see a reasonable amount of DNA editing. So we were pretty excited about this because we thought, well, maybe this is all just in cultured cells in the lab, but we thought maybe this will be a strategy for introducing these complexes into an actual tissue. Yeah, and this is just showing quantification of this. This is actually just showing that that there's no negative effect of introducing these peptides onto the protein. If you look at activity in vitro, just in a biochemical experiment, the modified and unmodified proteins have identical DNA cleavage activity. So this is it's a fully active version of Cas9. And so then the question is, can we do this in an animal? And I just want to show you one experiment here that I think is really, really intriguing. And so this is doing experiments in a mouse. So we're using injections into a mouse brain. So we can do these stereotactic injections. You're injecting half a microliter volume. And within that volume, using different concentrations of the gene editing complex. And again, we're doing this in the TD tomato mouse, so if cells get edited, they turn red. And so what you can see in these panels here, hopefully, is that as we increase the concentration of Cas9 that's injected, we see more and more cells that are red in these experiments, and we can quantify this. And furthermore, what was very interesting, I just want to point this out, that you know, you're injecting the same volume in every experiment, but the concentration is changing. And so what we found very interesting was that, you know, you get a larger volume of cells edited as you increase the concentration of the editing molecules. You're not changing the volume that's injected, though. So there's something interesting, I think, going on about the way that these injected molecules are actually trafficked in the tissue that we don't understand yet. And this is just some quantification down here of the amount of editing that we're seeing. And then here's another thing that I think is really interesting, sort of pointing to some potentially new biology, and that is that we see evidence for cell type specific gene editing. And so I won't walk you through every panel here, but this is an experiment in which we are injecting into different parts of the brain, and then we can stain these tissue samples with different proteins that are markers for different cell types. And by then merging those images with the imaging that we do for this TD tomato reporter of gene editing, we can look at these merged images and figure out if these particular types of cells are actually being edited. And what we found was that while there was a lot of editing going on for these hippocampal and striatal neurons, we really see no evidence right now for editing of astrocytes. And why is that? We actually don't understand this yet. And one experiment that we have ongoing is to just figure out some very basic things, like are these cells able to take up this modified Cas9 at the same efficiency as these cells? Is it simply an issue of access into the cell, or is there something more subtle going on? These cells are all taking up the editing proteins, but for some reason these cells are more resistant to editing. So we don't know the answer to that yet. So this is kind of just a summary of this work, and we're very excited to expand this now, and we just hired actually a great former postdoc from UCSF who's an expert in stereotactic injections in these mouse experiments to help Brett with some of the experiments that he's doing in animal models, and we, of course, are very eager eventually to test this in a mouse model of Huntington's. So in the last few minutes, I just want to turn to another aspect of CRISPR biology. So, you know, we've been really intrigued, as many people have, with the fact that there are so many different flavors of these CRISPR pathways. And what do they do? Do they all do the same thing, or do they do different things? And one of the things that's interesting is that while most of these pathways are targeting double-stranded DNA, there are two systems that actually naturally target RNA. One of them is called type 3, and this is one of these multi-component, multi-protein systems. And the other is called type 6, which involves a single enzyme, kind of analogous to Cas9, an enzyme called C2C2 that Eugene Koonen's lab a few years ago predicted would be an RNA-guided RNA-targeting protein based on its homologies to other sort of RNA cutting enzymes. And so several people in my lab started to play around with this enzyme. And I just want to show you a couple of things that we've learned recently about its behavior. And the bottom line is that this protein turns out to be a dual RNase. It's a protein that has two different RNA cutting activities. And the first one is an activity that allows this enzyme to produce its own CRISPR RNAs. And so what I mean by that is, so this is over here is just a phylogenetic tree. So Alex, Mitch, and Spencer in the lab started purifying these different examples of the C2C2 enzyme and testing its activities, again, in biochemical experiments. And what they found out, first of all, was that this protein binds to RNA molecules that are produced from the CRISPR array and cuts them in a very accurate way to produce the guide molecules that are used by the enzyme. And so this is an experiment where we start with uncleaved CRISPR transcripts, so these are called pre-CRISPR RNAs. And when we in the lanes on the right where we add the enzyme, in each case we see a product that corresponds to the mature CRISPR RNA molecule that can guide this enzyme to recognize particular nucleic acid targets. So it has that activity. And then it turns out that once it loads itself with its guide RNA, this protein is an RNA targeting enzyme that has a very interesting and different behavior compared to Cas9. So whereas Cas9 finds RNA sequences and cuts them very precisely at a particular place. This enzyme, once it finds a target RNA sequence that matches the guide RNA right here, it turns on a general RNase activity that allows it to cut up any RNA that's in the general neighborhood. And so the idea was that maybe this is essentially kind of a suicide protein, an enzyme that gets turned on upon detecting a foreign transcript in a cell and allows it to cut up any RNAs that are in the vicinity. And I'll just show you one experiment that shows kind of dramatically how this works. And so in this experiment, we're using one purified version of this protein, C2C2, and we add a radio-labeled RNA molecule called substrate A that migrates on a gel right here. And if we have no CRISPR RNA in the reaction, nothing happens to the RNA, but if we add the CRISPR RNA, now this substrate that has a matching sequence gets cut up. And you can see that it gets cut at multiple places, and if we wait long enough, it's cut up completely, right? It's completely destroyed. Now, if we do this experiment again, but we do it using the same CRISPR RNA, but we have a radio-labeled RNA that doesn't have a matching sequence to this RNA here, so it's substrate B, nothing happens. But if we dope in a little tiny amount of unlabeled RNA A that matches the CRISPR RNA sequence, the enzyme gets turned on, and now it cuts up molecule B, even though it doesn't have a matching sequence, right? So it's really a cool, kind of interesting activity. And we started thinking, how could we, is there any use for this? Is it a tool, or is it just kind of a curiosity? And it was really in a conversation with Jamie Kate at Berkeley that we realized that Jamie said, gee, you could probably use this as a very simple, low-cost way of detecting a transcript in a mixture, using a kit that's sold commercially by companies like Thermo Fisher for detecting ribonuclease. And the way these kits work is they sell you a little RNA oligo that has a quenched dye pair in the RNA, such that when this gets cut by ribonuclease, the dye molecules are released and they give off a fluorescent signal, so you can detect the presence of ribonuclease. And so what Mitch O'Connell and Alex Seletsky did was to test this in the context of a complex mixture of RNA that contained different concentrations of a target sequence that we wanted to detect. And without walking you through all these different combinations, what we found out was that detection was highly specific for the target matching the CRISPR RNA, and furthermore, we could detect down to about 1 picomolar of RNA, which is a pretty low concentration, and no PCR amplification or anything like that was required. So we've been thinking about how we might actually deploy this as a very simple kind of diagnostic method, and we're also working now with a couple of collaborators to test this as a way of marking or visualizing RNA transcripts inside of cells. And this is just a summary of how this family of enzymes operates by producing its own guide RNAs and then using a separate, different RNase activity to cut up transcripts only after being activated by detecting a particular RNA sequence. So in the last just like 1 or 2 minutes, I just want to finish by circling back to this sort of bigger question of, what do we do now that genomes can be edited relatively easily? And I really started thinking about this a lot as this technology was taking off over the last few years. And of course, lots of applications in stem cells and animal models of human disease, exciting opportunities to maybe actually move this into patients at some point. And you know that already there are ongoing clinical trials in China and soon to start here in the US to use the CRISPR-Cas system for gene editing in cancer patients to try to trigger activate the immune system against cancer cells. And then, of course, lots of experiments going on in many other kinds of organisms, plants, fungi, various kinds of animals. I love to show this picture here from a student at New York University in Claude Despon's lab who sent me this picture a couple of years ago. I love it. It's just, he was super excited because he was studying butterflies, and he finally had a way that he could manipulate the DNA in these butterflies to change the wing pattern and figure out, sort of the genetics of wing patterns in these animals. And lots of cool examples of this, where CRISPR is being used to really open up systems that previously were not genetically tractable and make them possible to study. And then, of course, lots of things going on in agriculture. This was a recent article in Nature that pointed out that mushrooms that were generated in an academic lab at Penn State University were made by making a knockout of a particular gene that prevents these mushrooms from turning brown when you cut them open. And the U.S. Department of Agriculture decided that this type of gene editing would not trigger GMO labeling of these kinds of products because no foreign DNA was introduced into these organisms. So this has triggered a very vigorous debate in various parts of the world because the way that GMO is defined is different in different parts of the world, and I think many regulatory agencies right now are really grappling with this and how to deal with this because there are many agricultural products that are now in the pipeline at different companies and in academic labs that involve this type of knockout gene editing. And then I just want to close by circling back to this question, and we talked about this a lot earlier today in various contexts and with the students. I think one of the powerful applications of gene editing, and certainly of this CRISPR-Cas9 technology, is in the germline. And this is a picture of a mouse embryo that's held by a pipette with a needle injecting DNA editing molecules into the cell. This was work that was done at Berkeley by Russell Vance. And of course, this has been done not only in mice and rats, but also in many other organisms, but also in monkeys. And it was really the work published in monkeys in early 2014 that really, for me, was kind of the final trigger that made me realize I really have to get involved in the ethical conversations that need to happen around this technology. And this has triggered a lot of interest in the media. There was this is taken from a cover of The Economist last year that was under the banner Editing Humanity, and people thinking about ways to use germline editing not only to mitigate disease, but also maybe for enhancement, and should we go there? And I don't know the answers, but I think it's a fascinating time right now in biology. I think all of us here have an opportunity to contribute to this discussion and to help educate people about what this technology really means, what it enables, and really to think together about where we want to go with it. And I just want to close by acknowledging a great group. So, science for me has always been highly collaborative. I have a wonderful group of students and postdocs. Some of them are shown here at a recent ball game. And we've also had great collaborators, and I can't list them all or I would have pages of slides, but I want to point out, of course, the work that we did with Emmanuel and Chris Chylinski, also with Jean Yo and his postdoc David at UC San Diego, and Anirvan Ghosh up until recently at Roche Biopharma, who really helped us get involved in the work that I showed you with editing in neurons. And then finally, many colleagues at Berkeley, but in particular, Eva, Jamie, and Bob Tejan, who have been incredibly helpful and fun to work with on various aspects of all of this. Couldn't do anything without funding, and I just want to acknowledge all of these groups. This is actually a P50 grant funded by the National Institutes of Health, but really it was the National Science Foundation back in probably around 2010 or so that gave me a very small but critical grant that allowed me to hire one of the first people that started working on CRISPR systems back before anybody had heard of them. And they trusted us with their money, and I'm incredibly grateful for that support. And thanks to all of you.
We'll take time for a few questions. We have a microphone here that will be passed to you. So first question right over there.
Hi.
Hi, Jennifer. Great talk. I'm curious about the C2C2 protein in bacteria. It seems like it must be regulated to be specific to its target. Otherwise, the bacteria with an active C2C2 would die, and it would never lead to a CRISPR tandem repeat formation. So it must be regulated in the actual cell where it's used, and have you explored that?
Yeah, I think it's a really interesting question. I mean, I think the idea that we're working on right now is that there's either active regulation or it's regulated just because of low-level expression. And so we're sort of, in the process of doing experiments in bacteria where you control the level of gene expression of these proteins and then investigate what effect that has on cells. We know that from our work and Feng Zhang's work that if you overexpress these proteins in bacteria, the bacteria are very sick or they die, so that's pretty clear. But the question is what happens when you dial back to a more physiologically relevant level of expression? And we don't know the answer yet, but I think you're absolutely right. And the other thing that's curious about these systems is how they acquire spacers in the first place, right? Because if they're targeting RNA, viruses, or transcripts, that's one thing, but how do they actually integrate the sequence that they use to generate that RNA in the first place at the level of DNA? No idea yet how that works.
Thank you. That was a really good talk. I have a comment and a question.
Yeah.
The comment is I have to take a little bit of an issue of having no baldness and a high IQ being placed on the same slide of the baby there. Anyway, there was a joke. The second one was how do bacteria deal with having the CRISPR sequence? Why doesn't that cut their own genome? How do they deal with this problem?
Yes, that's a great question, and I didn't explain that, but it's actually the purpose of the PAM sequence. So in the bacterial chromosome, when the new sequence from a virus is integrated, it's flanked by copies of these repeat elements that really specifically avoid the presence of PAMs. And we know that fundamentally for recognition of a target site, the PAM is essential. So without that, those sequences, even though they match the CRISPR RNA, they're invisible to the system. Yeah.
Great talk. How long do the bacteria keep the sequence? So how long does the immunity last, or is it very versatile, or is it constantly basically expanded with new viral sequences? Yeah,
so Jill Banfield has done a lot on that, looking sort of at populations of bacteria, and what her data suggests, and other people have looked at this, but I think Jill's done really the most probably in sort of wild, non-cultured bacteria. Her data suggests that certainly in some bacteria, they are actively acquiring, but they're actively losing spacers as well. So, and there's a polarity to these arrays. So their new sequencers are being acquired on one end, and they're being lost from the other end. What controls the length of the arrays in different bugs? Not known, and also how presumably it's, I guess it's some kind of recombination mechanism that allows for loss, but that is really not understood yet about how that dynamic works.
I have a couple of questions for you. If I understood you correctly, you thought that maybe RNPs could be used to more efficiently target specific cell types rather than viruses or other things, which was a statement, maybe I misunderstood it. It surprised me a little bit. And I wonder if you can say a little bit more about the strategies that might be deployed there.
Yeah. So I'm not sure about efficiency, because I think certainly viruses, AAV vectors and things like that can, certainly can get into cells very, very efficiently. I think we've been thinking about this more from the perspective of safety and cell type specificity. So, viral vectors, again, they can have tropism that you can take advantage of for selective delivery, so that's one thing. But the thing we really like about this RNP strategy is that you're not introducing foreign DNA. And so because of that, you don't risk integration events, sort of spurious events. That's one thing. And the other is that the lifetime of these particles is, depending on the cell type, but typically around 24 hours. And that means that after that time, a lot of these particles are destroyed. And so you really limit off targets because you're not continually expressing these editing molecules over many days or weeks. So and we definitely see evidence for lower sort of levels of detectable off targets when we use RNPs compared to plasmid delivery, for example. And in the future, where we're going with this in the longer term is we really are excited about the potential for tissue selective delivery. And so in a project I didn't have time to tell you about today, We've been working on modifying the Cas9 protein with ligands, chemical ligands, that are recognized by specific types of receptors. And we're doing this right now on liver cells and in the liver, but we eventually want to do this in other types of cells as well. Because then you could really have, cell type specific uptake, which I think would be amazing if one could get it to work. And right now it seems a little bit like pie in the sky, futuristic, but I think it's coming. And I think there will be probably different ways to do it in the future, and there are companies and other labs have various strategies for this. But I think this is a strategy that I think will be useful.
This is very exciting, I think. So the other question I had is related to the ethics. I mean, you kind of opened the door to this very broadly.
Yeah.
But I wonder if you can tell us just a little bit more where you stand. Are there certain absolute limits where you would say this is a no-go, or are you open to any possibilities, or can you give us a bit of a hint there?
Yes, and the thing is, that this has kind of been a fascinating kind of personal evolution for me as well, because I found my own views have been evolving. When I first started thinking about this, I have to admit to you that I really thought, germline editing for clinical use, we shouldn't go there, And I had various, reasons for thinking that. I thought, probably unsafe, and we probably don't really need it for most things, and there's sort of opens the door to a slippery slope, and all those sorts of things. But over the last couple of years, I've just met a lot of really interesting people that have helped to, sort of educate me about different aspects of this. I learned that in Israel, the government, so one of the arguments was, access, right? And we talked about this today with students. I found out that in Israel, the government actually pays for couples to have up to 2 kids by in vitro fertilization if they so choose, and furthermore, to use various kinds of, pre-implantation diagnostic selection that allows them to screen out embryos that might have certain genetic defects. So it's not that much of a leap from that to say, well, then you might also allow couples to introduce changes that would be beneficial or healthy for their kids, and maybe the government would decide to pay for that, or maybe insurance companies eventually decide that it's more cost-effective to do that than to have to treat someone for their entire lifetime with a chronic debilitating disease. So there's sort of that aspect to it. And the other thing is just practical. I mean, this is coming, there's no question. I mean, this is absolutely coming. I've been in discussions with people that are starting companies thinking about this for the future, to be able to offer this to couples, in the future. I've been in discussions with people in other countries that are moving in this direction. I think that the fact that now there's at least now 4 countries that allow CRISPR-Cas9 gene editing in human embryos for research purposes, up to the 14-day. And I think assuming that those experiments proceed and look promising, there will be increasing pressure to use it clinically. And so I really think this is, coming in the future, and that we need to just grapple with it and think about how we're going to use it responsibly. Rather, I just don't think it's probably realistic to say we're never going to do this. So.
Speaking of family and children, I will have one more question and then we'll.
Okay.
So what do we know about post-translational modifications of Cas9 proteins?
Of the Cas9 protein, post-translational modifications, I don't know anything about that. And if you know something, I'd love to hear it, but nope, we don't know anything about that.
Oh, sure, one more.
So most of the diseases that you might want to fix with Cas9 are going to be loss of function mutations in a reading frame, and that will require a DNA template to fix.
Yeah.
And then the RNP strategy becomes kind of less powerful. Have you
I
thought
think
about how
it
to
becomes
deliver?
more powerful.
How
And
do
the
you
re-
deliver the DNA at the same time?
Well, I think it becomes so at least in laboratory settings, which obviously is not, clinically relevant. Maybe we know that we can actually co-deliver templates for repair and see really pretty high levels of homology-directed repair. And so we actually started doing this first with Alex Marson here at UCSF and doing it in primary human T cells. And seeing really, quite robust levels of homology-directed repair by co-delivering these. The
Is that
challenge
with
is
nucleofection?
Say again.
With nucleofection.
With nucleofection, right? So the challenge is how would you do that clinically, right? And I think, you know, we're currently just at the early stages of thinking about this, but I'm excited about possibilities of either introducing base pairing interactions between a template DNA and the guide, end of the guide RNA that would allow sort of non-covalent association that would allow them to be co-delivered in this sort of a strategy that I showed you today, or potentially by other ways of transiently coupling the DNA templates to this protein RNA particle. So that's something that we're, again, just at very, very early stages of exploring. One could also think about, you know, I think recently there was a paper using RNPs together with AAV vectors for template delivery that looked interesting. Again, that was done in a cultured cell setting, but you could think about ways that you might use that clinically. So I think the opportunities there are actually quite interesting. And I'm also intrigued by work of Geraldine Seydoux at Johns Hopkins, where she's been using very short DNA oligonucleotides, single-stranded DNAs that are tiled across to create a larger sequence for integration by homology-directed repair, and getting some really interesting, pretty robust rates of repair that way. So again, you have to couple all of that with how you would deliver. But I think the pieces are all there. You can kind of see how you might be able to put this together. So.